Good morning. Myself Tejashree Pandhare. I am a student of final year B Pharmacy in Shivajirao Dhundre College of Pharmacy. My topic is bioavailability and bioequivalence, guided by Dr. Nitin Mohire sir as the principal of our college. So, content includes introduction, definition, objectives, importance, guidelines, design and conduct of studies, documentation, and conclusion. Introduction. It is essential to ensure uniformity in standards of quality, efficacy, and safety of pharmaceutical products. Reasonable assurance is to be provided that various products containing same active ingredient marketed by different licenses are commercially equivalent and interchangeable. Release of an active substance should be known and reproducible. Both bioavailability and bioequivalence focus on release of drug substance from its dosage form and subsequent absorption in circulation. Similar approaches to measure bioavailability should be followed in demonstrating bioequivalence. Objective. It includes development of suitable dosage form for a new drug entity, determination of influence of excipient patient related factors and possible interaction with other drugs, development of new drug formulation of existing drug, control of quality of drug production, influences of processing factor, storage and stability, comparison of ability of drug substance from different form of same dosage form produced by different manufacturers. Importance. Therapeutic efficacy. Generic drug approval, interchangeability, quality assurance, affordability and accessibility, and guides drug development. Definition. Bioavailability. Bioavailability refers to the relative amount of drug from an administered dosage form which enters the systemic circulation and the rate at which drug appears in the systemic circulation. Bioequivalence of a drug is achieved if its extent and rate of absorption are not statistically significantly different from those of the reference product when administered at the same molar dose. Supra-bioavailability. It is a term used when a test product displays an appreciably larger bioavailability than the reference product. Pharmaceutical equivalence. Pharmaceutical equivalents are drug products that contain identical amount of identical active drug ingredient, that is the same salt or ester in the same therapeutic moiety. Pharmaceutical alternatives are drug products that contain the identical therapeutic moiety or its precursor but not necessarily in the same amount or dosage form or as the same salt or ester. Therapeutic equivalents are drug products that contain the same active substance or therapeutic moiety and clinically shows the same efficacy and safety. Guidelines. US FDA guidelines on bioavailability and bioequivalence. FDA guidelines ensure generic drugs are equally to branded name version in absorption and effectiveness. Generic drugs must demonstrate equivalence through comparative pharmacokinetic studies, etc. Indian CDSCO guidelines for bioavailability and bioequivalence mandate bioavailability and bioequivalence studies for generic drugs to ensure bioequivalence with reference product evaluate the absorption and extent of generic drugs active ingredient to FDA approach, provide specific requirements, study design including sample sizes, duration and statistical analysis. European guidelines for bioavailability and bioequivalence mandate BA and BE studies for generic drugs to ensure equivalence with reference product, assess the drug's absorption, distribution, metabolism, and excretion to other global regulatory guidelines, etc. These are the parameters. Design and conduct of studies for bioequivalence and bioavailability studies are classified into four types: pharmacokinetic endpoint studies, pharmacodynamic endpoint studies, clinical endpoint studies, in vitro endpoint studies. Pharmacokinetic studies includes study design, study population, study criteria, conditions, and statistical evaluation. Study design. The basic design of an in vivo bioavailability study is determined by the nature of reference material and the dosage form to be tested. Study population. The number of subjects recruited should be sufficient to allow for possible withdrawals or removals dropout for the studies. Selection criteria. To minimize inter and intra-individual variation, subjects should be standardized as much as possible and acceptable. The study should be normally performed on healthy adult volunteers with the aim of minimizing variability and permit detection of difference between the study drugs. Women should be required to give assurance that they are neither pregnant nor likely to become pregnant until the study. Steady state studies where the drug has longer terminal elimination half-life and blood concentration after a single dose cannot be followed for a sufficient time, where the drug is likely to accumulate in the body. Characteristics of investigational during BA and BE study. Evaluation will be based upon the major concentration of the active drug substance in the biological matrix. Statistical evaluation. Statistical analysis should be in account of source of variation that are reasonably assumed to have an effect on the response. The logarithmic transformation should be carried out for the pharmacokinetic parameters C and AUC before performing statistical analysis. Various types of test designs that are employed are: first, completely randomized design; second, randomized block design; third, crossover and carryover design; fourth, Latin square design. Pharmacodynamic studies. Study in healthy volunteers or patients using pharmacodynamic parameters may be used for establishing the equivalence between two pharmaceutical products. These studies may become necessary if quantitative analysis of drug or metabolite in plasma or urine cannot be made under the sufficient accuracy and sensitivity, if the drug concentration measurement cannot be used as surrogate endpoint for the demonstration of efficacy and safety of particular pharmaceutical product. Clinical endpoint studies and comparative clinical trial. In the absence of pharmacokinetic and pharmacodynamic approaches, adequate and well-controlled clinical trials may be used to establish BA and BE. The number of patients to be included in the study will be designed on the variability of the target parameters and the acceptance range, and is usually much higher than the number of subjects in bioequivalence studies. In vitro studies. In certain situations, a comparative in vitro studies may be sufficient to demonstrate equivalence between two drug products. Biowaivers dissolution should generally be carried out under mild agitation condition and at physiologically relevant pH. More than one batch of each formulation should be tested. Comparative dissolution profile rather than single point dissolution test data should be generated. Documents required are signed detailed protocol, approval by ethics committee, volunteer information sheet, informed consent form, etc. Difference between reference and generic drugs. Reference drugs are expensive, takes 10 to 12 years and cost around 1 billion. Drug patent for 20 years applied before clinical trials begins. And in generic drugs they are 30 to 80% cheaper, fraction of cost of testing and development, and approved for sale after patent protection expires. Conclusion. Both bioavailability and bioequivalence focus on measuring the absorption of drug into systemic circulation. Establish that a new formulation has therapeutic equivalence in the rate and extent of absorption to the reference drug product. Importance of linking the commercial drug product to clinical trial material at a time of NDA, and importance for post approval changes in the marketed drug formulation. These are the reference. Thank you.